How confident are you with the concept of an action potential? It's something many biology students struggle to grasp, and if that's you, you've come to the right place. Action potentials are one of the ways our cells communicate. They allow neurons to transmit information throughout the body. This is important for a whole host of functions, including your ability to contract muscle, digest food, breathe and sweat, just to name a few. For example, let's say you touch the stove while it's still hot. We all know that there is an almost instant reflex to pull your hand away, and action potentials are absolutely essential for this process. When you touch the hot object, sensory neurons in your fingertips generate a signal which travels to the spinal cord, and then back through the motor neurons to withdraw your hand from the painful stimulus. And the thing that starts this whole chain of events is an action potential. Now, to explore action potentials on a deeper level, we should start by reminding ourselves of the three main structures of a neuron: the dendrite, which receives incoming information, the cell body, which processes information, and the axon, which carries information across the length of the neuron and to adjacent cells. Another important piece of information before we go any further is that a neuronal transmission is entirely dependent on the movement of ions across the cell membrane. Remember, ions are just molecules or atoms that carry a charge, either through the loss or gain of an electron. The two key ions we need to track throughout this process are sodium and potassium. When a neuron is not sending a signal, it is at rest. And when it is at rest, the concentration of sodium ions is higher outside the cell, whereas the concentration of potassium ions is higher inside the cell. This creates an electrochemical gradient across the cell membrane. Recall that with any type of concentration gradient, molecules will move from an area of high concentration to an area of low concentration, which means that if ions could move freely across the membrane, then potassium would flow out and sodium would flow back into the cell. The difference in total charge between the inside and the outside of the cell is called the membrane potential. A neuron at rest has a membrane potential of -70 millivolts. This means that the inside of the cell is 70 millivolts less positive than the outside. Ions can move across the cell membrane by passive diffusion through structures called ion channels. While some ion channels are always open, the ones which sodium and potassium are relying upon on this occasion require a signal. This signal is called the stimulus. Broadly speaking, once a stimulus is received, ions move and the membrane potential begins to shift away from the resting value of -70 millivolts. If the stimulus is strong enough to cause the membrane potential to reach a threshold value of -55 millivolts, an action potential will be initiated, triggering a series of events. So what exactly is an action potential? In simple terms, an action potential is nothing more than a temporary shift in the neuron's membrane potential caused by ions suddenly flowing in and out of the cell. The action potential can be separated into three main phases: depolarisation, hyperpolarisation, and repolarisation. Let's step through each of these. When the cell reaches threshold potential, previously closed sodium ion channels open. Remember, the sodium ion concentration was higher on the outside of the cell than the inside, so if those channels were to open, would sodium flow in or out of the cell? That's right, it would flow into the cell, as per its concentration gradient. As positive sodium ions enter the cell, the inside of the cell is getting more positive, and in effect, the membrane potential becomes less negative. It begins to approach 0 millivolts. This process is called depolarisation. The membrane potential will eventually become positive, and when it reaches about 30 millivolts, sodium ion channels shut and potassium ion channels open. Now remember, the potassium ion concentration was higher on the inside of the cell compared to the outside, so if those channels were to open, would potassium flow in or out of the cell? That's right, it would flow out of the cell, again in line with its concentration gradient. As potassium ions leave the cell, the interior of the cell becomes less positive and eventually negative. This is called repolarisation. We are returning to a state where there is a negative membrane potential. Now, these potassium channels are a little bit slow to close, and for a brief period, our membrane potential becomes a little bit too negative. This is called hyperpolarisation. You might hear action potentials being referred to as all or nothing events. This is because the threshold potential of -55 millivolts must be reached, or no action potential will occur and no neuronal signal will be fired. You can't have a partial or small action potential. The action potential either happens or it doesn't. So if we revisit the withdrawal reflex for a second, the object we were touching would need to be hot enough to be enough of a stimulus to reach the threshold potential and trigger the set of ion movements, which allows for an action potential to occur. But why is it so crucial? When the action potential fires, it releases neurotransmitters, or messenger molecules, which travel along nerves and tell us to move our hands. So there we have it, the action potential in just a few minutes.